Chapter 6: Lipids, Membranes, and the First Cells. In this chapter, you're going to learn about life's defining barrier, the plasma membrane. It's built of lipids and proteins, and we'll be looking at the lipid structure and function, and we'll see how lipids spontaneously form bilayers. We'll also see how substances move across these bilayers and categorize them as diffusion and osmosis. And lastly, we'll look at the role of proteins in the movement of these molecules across the plasma membrane. Okay, so when I say plasma membrane, I'm also referring to the cell membrane, and I can use these two terms interchangeably. But the bottom line is that plasma membranes serve as a selective barrier. That means they allow some things to go in and they keep other things out. So it facilitates chemical reactions necessary for life, and it allows chemical reactions to occur by sequestering appropriate chemicals. Lipids are the basic structure of the plasma membrane. So lipids are carbon-containing compounds, and unlike the other macromolecules that we've talked about, like proteins, nucleic acids, and sugars, there are no monomers for the cell membrane or for lipids. But the one thing that all lipids have in common, and this could be a test question, is that they are insoluble in water. And this insolubility is due to a high proportion of nonpolar carbon-carbon and carbon-hydrogen bonds. So when we talk about carbon-carbon and carbon-hydrogen bonds, we're actually talking about hydrocarbons. You know, like hydrocarbons are what you put in your car, and we call it gas, right? We look at the octane level. So it has a carbon backbone with hydrogen coming out from the sides. So we look at the basic unit of these hydrocarbons, maybe we can consider as an isoprenoid. Well, what is an isoprenoid? An isoprenoid is a hydrocarbon chain. That means that there's a carbon backbone and hydrogens coming from either side. And where do we see isoprenoids? We see them in pigments, scents, vitamins, and even as precursors for sex hormones. They're also the building blocks for more complex lipids, and the reason why this is important is because lipids are the major component of cell or plasma membranes. We also have fatty acids, and a fatty acid is a hydrocarbon chain, or like a small isoprenoid, but it has a carboxyl group at one end. Remember, carboxyl groups are functional groups, and it's COOH. So we find these at one end, and we can say that a fatty acid is either saturated or unsaturated. So what do we mean by a saturated or unsaturated fatty acid? First of all, I'm reminding you a fatty acid is just a hydrocarbon chain, carbons and hydrogens, with a carboxyl group at one end. If it's saturated, then it means that there's a lot of single bonds and it's saturated with hydrogens. In other words, it has a maximum number of hydrogen atoms. If it's unsaturated, then it means that there's one or more double bonds and it isn't saturated with hydrogens. So polyunsaturated chains have many double bonds, and these double bonds form kinks, and that's why they're not saturated. I love this example in figure 6.1. First of all, you see something called an isoprene. It's just showing you a small hydrocarbon, and if you add a whole bunch of isoprenes together, you get an isoprenoid. And if you put a carboxyl group at one end, then you get a fatty acid. So that's what you see in the middle. But notice in B, that fatty acid is saturated, and that's why it's very linear, because it's all single bonds and it has a maximum number of hydrogens. But if you look at the last diagram on the right-hand side, you see that there's a kink, and that kink is caused by a double bond. That means it's unsaturated. It doesn't have the maximum number of hydrogens, and you can tell by counting the hydrogens, or you can look at the double bonds. So saturation is important because it changes the physical state of a lipid. Foods that contain lipids with double bonds are said to be healthier. And usually if they're at room temperature, then they're liquids at room temperature. So they're said to be healthier for you. Okay, so saturated lipids are usually solid at room temperature. And saturated lipids with long hydrocarbon tails are stiff solids at room temperatures. So let me think of some examples of saturated and unsaturated fats. So I'm thinking of olive oil, canola oil, corn oil. All of those I have to buy in containers, and they're unsaturated fats. That's why they're liquids at room temperatures, because they have kinks. And if they have kinks, then the hydrocarbon chains don't lay densely packed to one another in a linear form. So that's why they're liquids, because of the kinks. And therefore they're less dense and they're a liquid at room temperature. But then I'm thinking of one of my favorite things is bacon and bacon grease. If I leave bacon grease in a pan, after a while it's going to become a solid at room temperature. So I know that it's saturated. Why is it saturated? It's saturated with hydrogen, and it's making the fatty acid tails linear. And because they're linear, you can pack more fatty acid tails together, and that's what's making it a solid. If it's a solid, it's very hard for things to go across that solid layer. I'm also thinking of earwax and beeswax. Okay, both of those are made of lipids too, and they're very solid at room temperature. I love figure 6.2 because it shows some examples. So when you see that zigzag, when you see the zigzag, what does it represent? It represents a hydrocarbon chain. You can imagine carbons at each one of those points and hydrogens being bonded to that carbon. And notice at one side there's a carboxyl group, COOH. So let's look at butter. Yum. Notice that butter is saturated. Notice it's all single bonds, it has a chain, and that's why it's solid at room temperature. Look at beeswax in the middle. It's even stiffer and more solid at room temperature than butter, and that makes sense. Look at how long the hydrocarbon chain is, and notice it's made up of all single bonds. I don't see any double bonds, and it's laying really flat, so that means you can pack a lot of these tails together, and that's why beeswax is solid at room temperature. And then at the very end you see safflower. Safflower has double bonds, and that's going to give us kinks. It's like me bending my elbows like this. No one can get close to me because my elbows are out like this. So it's less dense, and because of that, it's a liquid at room temperature. At the very bottom we have hydrogenation, and that's Crisco. I'm a Crisco kid. I grew up on Crisco, and a lot of people my age did too. But do I recommend you using Crisco? And the answer is heck no, because Crisco is artificial. Crisco is a solid that you can put in a storage cabinet, and you don't need to refrigerate it. You don't need to put it in a glass jar. So it's easy to transport, it's easy to sell in a cardboard container, right? So that's why companies made Crisco. But the truth is it's not very healthy for us. That's why it lasts so long in our pantry, because not even the bacteria like it, because the bacteria know that it's artificial fat. So for us gals that have more fat on us, we really should eat natural fats and not hydrogenated fats. Lipids do not possess shared chemical structure, so there is no monomer for fats. The only thing that they have in common is that they're insoluble in water. So they have a wide variation in structure, and there's no monomer for them. Many do have a hydrocarbon skeleton that can be put together in several different ways. So we're going to talk about 3 types of fats, but really the most important fat that we'll be talking about in this chapter are the phospholipids. We'll be talking about steroids, fats, and phospholipids. Okay, so what is a steroid? A steroid is actually made of a 4-ring structure, so it looks nothing like a fatty acid. So these 4 rings are attached together, and this is one type of a lipid. So what are some examples of steroids? They're hormones such as estrogen and testosterone. But another type of steroid is cholesterol. And cholesterol is really important because we can also find that in the plasma membrane. So we'll be talking about cholesterol in this chapter. Okay, so this is what cholesterol looks like. So notice most of it is hydrophobic. It has non-polar covalent bonds. Just one little bit of the end is hydrophilic. So this is the 4-ring structure, and this is what a steroid looks like, or what cholesterol does too. When we talk about fats, we usually are talking about triacylglycerols or triglycerides. If you know someone in your family that's gone to the doctor, they'll take a blood sample and they'll look at the fat content in your blood. One of my sons always tells me, "Mom, stop eating sour cream and fatty cheese because it's raising the triglycerides in your bloodstream." They go, "Isn't that gross?" And I go, "No, all I can think of is yum, yum, yum. I love cheese, I love sour cream." And I know I shouldn't say it, but it's the truth. So what is the role of fats that we find in our body? And the primary role is for energy. So a large number of high energy bonds, they're usually non-polar covalent bonds, so there's a lot of potential energy in those bonds. And so when those bonds are broken, they release a lot of energy. So I usually wear baggy shirts to cover a lot of my fat that I'm carrying around with me, but I guess I can say with pride that I'm carrying a lot of energy with me, just in case I need it. Fats are formed by dehydration reactions, and you guys know in a dehydration reaction, water comes out. Water comes out. So usually when we have a triglyceride, it's 3 fatty acids and they're held together by another molecule called glycerol. And this is through an ester linkage. You don't have to know the ester linkage for the test, just because fats don't have a basic monomer, so they don't have a basic linkage that can be used for all fats. So, but for fats, there is an ester linkage. Okay, so let's compare a carbohydrate that you find on top in figure 6.4 to a fatty acid down below. They sort of look the same, don't they? There's a carboxyl group at one end, that's COOH, and at the other end we have a carbon backbone, and for sugar, the bottom part is bonds with hydrogen, but on the top part above the carbon backbone, there's hydroxyl groups. For fatty acid, remember, they're just hydrocarbons. So there's hydrogen below and above the carbon backbone. That means there's more non-polar covalent bonds than there are in sugars. So pound for pound, there's more energy in a fat than there is in a sugar. And a lot of it just has to do by the number of non-polar covalent bonds. Because if electrons are shared evenly, they have more energy than those found in polar covalent bonds. Remember, oxygen is electronegative and it will pull electrons towards oxygen. And when it does, those bonds are not as strong as non-polar covalent bonds. The last type of lipid is a phospholipid, and this is the one that we'll be concentrating on in this chapter. And the reason why is phospholipids form cell membranes. So it consists of a glycerol linked to a phosphate and 2 hydrocarbon chains. So remember a triglyceride? A triglyceride has 3 fatty acid tails. So if we remove one of those, and we replace it with a phosphate head, then we have a phospholipid. Okay, With fatty acid tails, these are found in domain bacteria and eukarya. If there's an isoprenoid tail, then this is found in domain archaea. So this is one of the differences among the different types of cell membranes. But the primary role of all phospholipids is to form the cell membrane. So I love figure 6.5 because it shows you a triglyceride. And remember, a triglyceride is 3 fatty acid tails held together by a glycerol and that ester linkage. If you remove one of the fatty acid tails and replace it with a phosphate group, then you have a phospholipid that you see on the right-hand side of your screen. And this is what it looks like also, and another way of looking at it. So it says here lipids have a wide array of functions, and some of these functions include to store chemical energy, to act as a pigment that captures sunlight, serve as signals between cells. It can form a waterproof coating on your skin and cells. It can also act as vitamins too. So many lipids can contain both hydrophobic and hydrophilic regions. So you guys remember what hydrophobic and hydrophilic mean. Hydrophobic is water hating, hydrophilic is water loving. So the head region with the phosphate group is hydrophilic, and the tail is hydrophobic. So the phosphate group has a charge to it, and this is what's making it charged. Remember, birds of a feather flock together. Water's charged, and the phosphate head is charged, so it's hydrophilic. They'll react with each other. But with nonpolar covalent bonds, the hydrocarbon tail will be hydrophobic. So we call that, when there's both hydrophilic and hydrophobic regions, we call that amphipathic. So phospholipids are amphipathic. They have both water-loving and water-hating regions. Another example would be cholesterol. So the plasma membrane is amphipathic. It has both water-loving and water-hating regions. So it makes for a great barrier. Amphipathic lipids do not dissolve in water, but the hydrophilic heads do interact with water and the tails do not. They move towards the inside and the heads move outside. So when that happens, then they spontaneously form these things called micelles. So what are micelles? They're like little tiny bubbles. They're like little tiny fat globules that you can see floating in water. So now you know why when you have like a vinegar and oil dressing that you see tiny little globules or vesicles of oil floating around in the vinegar. And this is because of the water hating and water loving regions. So here's an example of a single layer micelle. So notice that the phosphoheads, which are water loving or hydrophilic, face out. Notice that the tails that are hydrophobic face in. And these micelles form spontaneously when you place them in water. And below you see a lipid bilayer. The same thing happens. The heads automatically face out because they'll interact with water, and the tails that are hydrophobic will face in and they will not interact with water. Phospholipid bilayers. Phospholipid bilayers consist of phospholipids and they form spontaneously in water. And that means when they form spontaneously that there's no energy input. Okay, so if they form spontaneously, in most cases, that means that entropy has decreased. Okay, so what are we talking about? Entropy is disorder. So spontaneous processes tend to increase entropy, and yet when these phospholipid bilayers form, just the opposite occurs. There is more order. And this is due to something very unique. It says a cage of water forms around this lipid aggregate. It's like what you see when you see a salad dressing made of vinegar and oil. Another thing that I'd like to think about too is, when there's an oil spill in the ocean, how come it just doesn't dissolve and go away? You have that oil spill, and that's because there are hydrophilic and hydrophobic regions to that, and mainly because the oil is made up of long hydrocarbon tails. It's basically nonpolar and it's not going to interact with water. So this is what we're talking about. So it says here vesicles can be formed from phospholipids just because of these two regions, the water-loving and water-hating regions. So they form these bubbles, and we've often seen these, like I said before, when we have oil, something oily in water. And we can also create artificial ones, and these are called liposomes. I used to have a supplemental instructor help me out, and when he was working on his master's in chemistry, he actually focused on liposomes, and he was using these like little fat vesicles or vesicles made out of lipid actually as a carrier to carry like medicine within a body. And I thought at first like, nah, that can't happen, but definitely there's been a lot of research on these liposome, these artificial lipid membranes. And here you see the hydrophobic and hydrophilic regions of a liposome. Okay, so these artificial membranes actually have different degrees of permeability. So what do we mean by permeability? And that's the ability for molecules to actually pass the membrane. Can all substances pass through the membrane, or is there selective permeability where some things can cross and other molecules cannot? Okay, so we use these artificial membranes to actually evaluate the movement of different kinds of molecules across a membrane. And here in this diagram, you can see that there's a beaker, and this beaker has a septum, which is a fancy word for wall. And in the middle of this glass wall, you see that they have placed this like lipid bilayer, this artificial membrane. And then if you look on the right-hand side of the screen, you see that they've put in all kinds of molecules indicated by the different sizes and different colors of spheres on one side. And the question is which ones can pass? Can all of them pass, or do they pass at different rates? How is it that some molecules can move across a membrane? So we know now that phospholipid bilayers have selective permeability. That means that some molecules can cross while others cannot. So usually small nonpolar molecules can move across the phospholipid bilayer very quickly. Why is that? Does that make sense? And it does, because small means you can move through in between the tails very easily, and if it's nonpolar, there's no charge. And the phospholipid tails make up most of the phospholipid membrane, so it makes sense that a non-charged molecule or a neutral molecule could move through more rapidly than one that is charged. So what are some examples of a small and nonpolar molecule? Like oxygen would be an example of that. So oxygen is a small nonpolar molecule that can move quickly across a bilayer. On the other hand, glucose is large, and it takes glucose is too large to move across the membrane. It might need some help. I love this figure 6.9 because this shows you and gives you examples of some molecules that can move across the phospholipid bilayer. So remember, if molecules are small and nonpolar, they can move across, and this is indicated by yellow. Then there's other molecules that are small, but oftentimes they have a charge, and sometimes it is hard for them to move across. And notice that water is one of those. And then we get to larger molecules like glucose and sucrose. They're large, they don't have a charge, but they still have a hard time moving across the phospholipid bilayer. And then lastly, at the very end, we see really, really tiny atoms. And notice they can't move across at all. And the reason why they can't move across is because they're charged. And most of the phospholipid bilayer is nonpolar. The whitest part is made out of the fatty acid tails. So how can something charged move across this non-charged or charged neutral region? So that is something that's really interesting. How do lipid structures affect membrane permeability? Not all phospholipids are the same. So what are some of the factors of the phospholipids that affect the selective permeability that influence why some molecules can cross and others cannot? So we have to look at the length of the hydrocarbon tails. We have to look at the saturation state of the hydrocarbons, whether there's a lot of single bonds or are there double bonds in the fatty acid tails. And we also need to look at the presence of cholesterol. Remember, cholesterol is a steroid and it's made out of a 4-ring structure. So let's look at the degree of saturation and length of tails and how it affects the behavior of the membrane. If the tails are unsaturated, that means there's kinks, then you can see that it's not dense, the tails are not densely packed. And if the tails are not densely packed, then we can imagine that a lot more molecules can cross the membrane. If the tails are saturated, that means they're all single bonds. And there's very few spaces in between the tails because they're more densely packed to one another, like sardines in a can. Then it'll be very hard for molecules to squeeze in and move across the layer. Hydrophobic interactions will become stronger, and this will actually decrease selective permeability. Membranes containing phospholipids with even longer tails will be even less permeable. I love figure 6.10 because it just shows what we just talked about. So if you look at lipid bilayers with short unsaturated tails, we have more molecules being able to cross. And that's because the phospholipids aren't so densely packed. There's like pathways for molecules to move across. But if the fatty acid tails are saturated, that means they're all single bonds. They're going to lay straight and they're going to be densely packed. And if the tails become longer, then it's going to be very, very difficult for molecules to find a pathway to move from one side to another. Cholesterol does affect membrane permeability. Cholesterol molecules present in membranes are present in membranes, but the more cholesterol there is, it increases the density and hydrophobic sections. And when that happens, it reduces permeability. So the more cholesterol you see in a membrane, the less permeable it is to molecules moving across. In this experiment, what they did is they looked at temperature and they looked at the amount of cholesterol found in a phospholipid bilayer. So if you look at the graph at the bottom of the slide, you'll see that the x-axis is temperature in degrees Celsius. If you look at the y-axis, it's the degree of permeability. So if you look at the blue line, you see that it has the most permeability. That's a cell membrane with no cholesterol. So as temperature increases, notice it increases. In other words, it curves up. Notice as we start increasing the amount of cholesterol, it's going to decrease the amount of permeability. And notice in all three cases, whether there's no cholesterol, 20%, or 50% cholesterol, notice as temperature increases, so does the permeability of the membrane. So this is a very interesting graph. How does temperature affect the fluidity and permeability of membranes? As we saw in the previous graph, as temperature increases, so does permeability. So the opposite is true too. Membrane fluidity will decrease as temperature drops, and as the temperature drops, so will permeability. I always think about, like solid bacon grease in a pan. If you were to pour any kind of solution on top of that solid bacon grease, would it cross all that fatty layer? And the answer is no, and we wouldn't expect it to. So we intuitively know this. Figure 6.12 shows that actually the phospholipid bilayer is always moving around. When I was really young and someone was instructing me decades and decades and decades ago, like what does a phospholipid bilayer look like, I always used to think that it looked like cellophane or plastic wrap, that it was this thin sort of hard layer that was surrounding a cell membrane, but really in reality it's like a glob of oil, and these molecules are always moving. And what this figures show is that there's a lot of lateral movement in the phospholipids, but it's very rare that a phospholipid from the top layer will move to the bottom layer, but there is a lot of lateral movement. Now let's talk specifically about the movement of molecules moving across the phospholipid bilayer or the plasma or cell membrane. So there are some words that we need to know. And so it says here, dissolved solutes are in constant random motion due to their own thermal energy. And we can see this in Brownian motion, that there's this like shaky movement of all molecules. And as they shake and bounce off each other, then this random motion, then they can move out or they can be used to cross a membrane. So one of these movements is called diffusion. So what is diffusion? It's spontaneous movement of molecules in ion, and it moves along a concentration gradient created by the difference in solute concentration. So there's a net movement from a high concentration to a low concentration. Oftentimes I think of molecules like balls on a billiard table. When you put them all together within that triangle, they're closely together, but when you put the cue stick and you hit them across, then they move across in a random motion. But we don't need a cue stick for molecules because they have their own thermal energy, and that thermal energy, they start bouncing off each other and start shooting away from one another. And so diffusion is that movement from a high concentration to a low concentration. So I always think of actually Saturday Night Fever. John Travolta going up and down, high to low, high to low, high to low. So the diffusion is moving across this concentration gradient. Diffusion along a concentration gradient increases entropy. And what is entropy? It's disorder. So we hit equilibrium. So equilibrium occurs when molecules or ions are randomly distributed throughout a solution. So I can think of several things. Like you could drop like blue dye in a glass of water, and at time zero you see a great big blue glob. But then as time increases, isn't it true, the whole glass of water becomes a light blue. So that means it's hit equilibrium. So this takes no energy, it just moves across randomly because of that thermal energy that molecules have. And this is called passive transport. What is passive transport? Passive transport occurs when substances diffuse across a membrane in the absence of any outside energy source. So there is no additional energy source, just the thermal energy from each of the atoms or molecules. In this example here, you see a lot of particles in solution, and they're in these beakers, and these beakers all have septums, and there's an artificial membrane. Okay, so let's look at the very top one. If you look at the top beaker, there's white circles on the right hand side, and on the left hand side you have these purple triangles. So what's going to happen over time? Over time, if we look at these gradients going from high to low, high to low, then we see that the triangles will diffuse from the left to the right hand side, and the white circles will move in the opposite direction. So this is diffusion. It takes no added energy, and eventually it will hit equilibrium. There's no net diffusion between both sides of the beaker. Water moves quickly across lipid bilayers, but there's a special case of diffusion, and this is called osmosis. So what is the definition of osmosis? It's the diffusion of water. Water is so special it deserves its own special definition, and that is osmosis. It too moves from a high to low concentration, high to low, just like John Travolta. So it moves from a high concentration to a low. Now, I used to make it very difficult to determine how osmosis and how water moved, but I had a supplemental instructor, a different gentleman, and he said, you know, a simple way of explaining it, water always follows the solutes. It always follows the salts or the molecules dissolved in water. So that way you can tell which way water is moving. When we talk about the movement of water, we also talk about something called tonicity. That means if there's movement of water, then there must be a different concentration of molecules on either side of the membrane. So to describe that, we have hypertonic, hypotonic, and isotonic concentrations. Hypertonic means that there are more molecules outside of the cell than inside. In a hypotonic solution, there are hardly any molecules outside of the cell relative to what we find inside of the cell. And in an isotonic solution, there's about an equal amount or equal concentration of solutes or molecules both inside and outside of the cell. So the question is how does this affect the movement of water? In this case here, we see osmosis in action. So here we have molecules on both sides of the membrane. And notice if we look at the beaker up on the top, we see that we have these green spheres indicating these molecules that are in solution. But what we should be told is that these molecules are either too large or maybe they're charged and they just can't cross the membrane. So to equalize everything, the only thing that can move is water. Since the molecules can't move, these green molecules, they can't move, the only thing that can move to create equilibrium is the water. So if we look at the beaker below, water is moving. It's moving to the side that has the higher concentration of molecules. Remember what I said before? Water follows the salts. Water follows the molecules that are dissolved in water. So notice water moved from the left side of the beaker to the right side. So now the concentration is about the same. This is pretty fascinating, right? To see a rise in the water level, and this is due to osmosis. Water follows the molecules. Water follows the salts. In a hypertonic solution, let's say you drop a red blood cell into a beaker of water, a hypertonic solution, that means that there's more molecules outside of the cell than inside. This will cause water to move outside because water follows the salts. And when that happens, the cell is going to shrivel up. In a hypotonic solution, if you were to drop a red blood cell into a beaker of DI water that has no molecules in it, that means there's more molecules inside of the red blood cell. Water follows the salts. So water will come rushing into the cell and the cell will begin to swell. In an isotonic solution, there is no net movement of water because there's the same concentration of molecules on both sides of the membrane. I love this diagram because this shows the different types of tonicity and how the movement of water. This is so important because this is how our kidneys function. This is how we brine something in the kitchen, or this is why we have IVs that have a little bit of saline solution in them. All of this is very important to life itself. So in the first example, we have a hypertonic solution. Notice that there's more molecules outside of the cell, and it's going to force water to move out and therefore shrink the cell. In the middle frames, you see that we have a hypotonic solution. There's more molecules inside of the cell. Water follows the molecules. So now water is going to rush into the cell, causing the cell to swell. And the cell can swell so much that it can burst. So that's why when you go to the hospital and they give you an IV, they give you an IV that has saline solution because they don't want your cells to burst. And then lastly, you have an isotonic solution. And this is where you see the same concentration of molecules both inside and outside of the cell. And because of that, there's no net movement in water. You can take a look at this video, and if it doesn't work in this presentation, you can go to the presentations that are just PowerPoint presentations and not videos. Okay, so membranes and chemical evolution. So what did the first cell look like, and were membranes important? So one of my mentors in my personal career was a chemist, and he was a very religious man, but he would always tell me, Naomi, imagine the very first cell and remember the probability of having a naked replicator, which is an RNA molecule, go inside of a membrane, which is a phospholipid bilayer. He goes, imagine what that probability would be. The probability is so low. That's why it took millions and millions and millions of years for this to occur. So maybe the first protocell was likely RNA, a naked self-replicator enclosed by a phospholipid membrane. And voila, this is the beginning of biological evolution, or the beginning of life itself. So this is what a protocell might look like. Here's a membrane, and inside you see RNA molecules. And in this membrane, it has to be that on the outside you have nucleotides, the building block of RNA that can go easily into the membrane to actually synthesize new RNA molecules. Proteins can alter membrane structure and function. So remember, selective permeability, right? Some things can move in and other things can't. But isn't it true? Don't we want things like glucose to go into the cell? So what do we do when molecules are too large? Or how do molecules move in that have a charge? So there has to be some way to facilitate that movement. And the facilitator is going to be proteins. Proteins embedded in the phospholipid membrane. So proteins can be inserted into the membrane, and just like phospholipids, they too are amphipathic. Remember, amphipathic means that there's hydrophilic and hydrophobic regions, and these hydrophilic and phobic regions are caused by polar, charged, or nonpolar regions in the molecule. So these proteins are embedded into the phospholipid membrane. Here you see in yellow and green those regions that are amphipathic, those regions that are water loving, and those that are water hating. So if it's green, it's water loving, and they're going to be facing the outside. If it's water heating, that means hydrophobic, and they'll be in the inside of the phospholipid membrane. This gives the term fluid mosaic model. So what does the fluid mosaic model indicate? It suggests that our plasma membrane is not just made of actually a phospholipid bilayer, but there's other things involved. It's much more complex. And adding to that complexity are proteins. So the mosaic is that it's not just one type of molecule, but different types. And one important part are these proteins. The fluid part is that lateral movement that we had talked about, this side-to-side movement of molecules moving all the time in the membrane. So a long time ago, that would be in the 1950s when I was born, ages and ages and ages ago, they knew that there were proteins associated with the plasma membrane, but they didn't know the architecture of it. Was it that on the top model that proteins were based on the outside and inside of the phospholipid membrane and it sandwiched the lipids? Or was it that they were transmembrane proteins, that there were actually proteins going all the way through the phospholipid membrane? Well, we know now it is the bottom model, the fluid mosaic model. So this is the model. This is actually how our membranes look like. It's the bottom model, the fluid mosaic model. So how do we know this? Is it just an idea or do we have evidence? So we have evidence using the scanning electron microscope, SEM. And the other thing is we've used freeze fracture methods to actually separate the phospholipid bilayer. And when it's separated, we see that there's a lot of pits. One side will have a lot of protein sticking out, and we'll have on the other side some proteins in pits. Well, what caused the pits? The pits must be caused by proteins going all the way through the membrane. And this is how we know it's the fluid mosaic model. So what do we call it when proteins go from one side of the membrane to the other? We can either call them an integral protein or a transmembrane protein. They both mean the same, and I can use these terms interchangeably. So integral and transmembrane mean the same. It means they're proteins that span the membrane. They're like doorways into the cell. Then we have peripheral membranes, and they're either going to be found on the outside or on the inside, but they don't go all the way through the membrane. Okay, now how can we study these membranes or these proteins that are found inside or going through the membrane? And we can use detergents. And these detergents can separate proteins from the phospholipid membrane. So we know from these studies using detergents that there's different kinds of proteins. And we can call these doorways either channels, carriers, or pumps. So here in this diagram, we see that detergents are represented by these like little red stars. And notice these little red stars sort of encircle the phospholipids and the proteins, and when they do, they separate them, and that sort of makes sense. I'm thinking about Dawn detergent. You know, that's how we release the greasy fat from a pan that we've been cooking from. Okay, so one of the first integral proteins or transmembrane proteins are channels or ion channels, and these are specialized doorways. So these are doorways that only allow specific ions to move through. So remember what ions are? They're charged atoms. They either have a negative charge or they have a positive charge. So instead of just forming a concentration gradient, they can form this electrochemical gradient. So it's not just a gradient with the chemical, a buildup of chemical on one side, but actually there's a buildup of charge on one side. And we'll see this in a future slide. So ions can diffuse down their electrical chemical gradient, but they have to move through this protein, this doorway into the cell. So you know what I think a channel is? It's like a doorway, but there's no door. There's just an archway, an opening, but no door that opens and closes. So here's an example of sodium and chlorine. Notice on one side of the membrane we see an abundance of sodium. So we have an abundance of sodium, which is a concentration buildup, but we also have a buildup of positive charges. And this charge and concentration gradient is going to move sodium from one side of the membrane to another. But it's going to move through this channel, this opening within the membrane caused by a protein. So it says here, cells have different types of pore-like channel proteins in their membranes. And all of these channels or openings are very selective. So there's a protein that just allows sodium to go through. There's another protein that just allows water to go from one side of the membrane to another. So they're very selective and they're openings. So here's an example of an aquaporin. In this particular channel made by a protein, a transmembrane protein, only water can move in and out of the cell membrane. Then we can have gated channels. Well, a gated channel means there's a gate that opens and closes to this opening. And it's usually there's some type of signal. And the signal is usually a change in the charge of the membrane. So a change in electrical voltage running across the membrane will either open or close a membrane. You know what this reminds me of? It's like when someone has a swipe card that can open and close like a door, or one of those gated arms that you see in a parking garage. So here you see that actually the channels can open and close. And notice on the left-hand side, positive charges are on the outside of the cell and negative charges are on the inside. But notice when there's a charge reversal, it opens up the channel. So this is an example of a gated channel, like a gate. And when the gates are open, notice that potassium, which has a positive charge, is moving across this protein through this channel. Movement of substances through channel proteins is passive. What does that mean? It doesn't require any outside energy. It's all based on electrochemical gradient. Ions and small polar molecules can diffuse across this lipid bilayer. So what we call this passive movement through a protein that acts as a doorway, we call this facilitated diffusion. I promise you this will be a test question. So please note diffusion and facilitated diffusion. Both types of diffusion are passive. They do not require additional energy. Okay, so what is a common disease that we can have when these channels are not functioning? And one such disease is cystic fibrosis. Cystic fibrosis is when you have a lot of mucus accumulate around all of your internal organs, especially your lungs. And if you have a lot of mucus around your lungs, you can imagine it will be very difficult to breathe. So people with cystic fibrosis usually don't live for a very long time. Well, what causes all of that mucus buildup? It's a defective channel. And this channel is specific for the movement of chloride ions to move in and out of the cell. So here they give an example of cystic fibrosis and when we have channels and when we don't. And this is what's causing mucus buildup. This is sort of fascinating, this connection between specific ion channels and a terrible genetic disorder. Okay, the next type of protein is a carrier protein. So remember, I liken all of these integral or transmembrane proteins to doorways. And this doorway is like a fancy doorway. This doorway can open and close and it moves. So that's called a carrier protein. And we use this, one famous one is the movement of glucose across a membrane. So it says here, a carrier protein selectively picks up a solute or a molecule on one side of a membrane and it drops it on the other side. So it's sort of like a moving doorway. So I use glucose. So a special carrier protein is called GLUT1. GLUT1 picks up a glucose on one side of the membrane and deposits it on the other side. So this is called a carrier protein because it's carrying the glucose across the membrane, but it is still a form of diffusion. I love figure 6.25 because you see how this carrier protein GLUT1 opens up on one side of the membrane, and notice how selective it is. It's only going to carry glucose across the membrane. When glucose fits in to the membrane, notice it's going to open on the other side. It's going to go through this conformational change, and it's going to drop it on the inside of the membrane. So this is called a carrier protein. So the last type of transmembrane or integral protein that we're going to talk about is active transport. And active transport is associated with pumps. So that will be our last kind of doorway into the cell. This doorway that is caused by a specific type of protein. So remember, facilitative diffusion is going through channels and carriers, and it's a form of passive transport. No additional energy is needed. Active is just the opposite. Things are moving against a concentration gradient. It's going from low to high. It's going to go from a low to high, low to high. It's like pedaling up the Franklin Mountains. It's going to take a lot of energy. So A is for active transport. A is for ATP, the energy used in cells to make things work. So that's why we call it a pump, because it's moving against a concentration gradient. I always think of that old-fashioned pump. To pump a tire, you have to work really hard. It requires energy to put air in a flat bicycle tire. So it says here structural changes can lead to active transport, and one of the most famous pumps of all time is a sodium-potassium pump. Okay, so our cells are always trying to pump out sodium, and they're always trying to bring in potassium. But the truth is we have to move both against their concentration gradients. What do I mean against a concentration gradient? That means if I took one of you students and I told you, come on, get on the bus as people are getting off the bus, and I have to push you onto the bus, I have to, because as students are getting off, I'm insisting that you get onto it. So you're moving against a concentration gradient. And that's what is happening with potassium and sodium at the cellular level. They're moving against their concentration gradients. So in this diagram here, you see that the green represents a pump. And this pump requires energy in the form of ATP. And you can actually see ATP. So ATP is providing the energy to pump out sodium and pump in potassium. So both of these molecules are moving against their concentration gradient, and for this reason they need additional energy. I hope that you can see this video with this on this movie, but if you can't, please close it and go to the presentations in the presentation folder, and you'll be able to see this presentation. Okay, so there's such a thing as called secondary active transport or co-transport. This will not be on the exam. I will be happy if you know passive transport, diffusion, facilitated diffusion, and active transport. So you don't need to know secondary active transport, but what it means is when you have active transport, these gradients often open the doorways for other molecules to move in and out of the cell. It's sort of saying like molecules riding on the coattails of others, so other molecules can move across when these doorways are being used. Plasma membranes define intracellular environment. So what does that mean? You know, I think of the best analogy is my own skin. If I take a look at my own skin, this skin makes me me. It's the barrier to me. And the inside of my skin, what's inside is totally different than the outer environment. I'm always at 98.6, and there's a lot of reactions happening inside me that are not happening outside in the environment. So membranes are very important. So biological membranes allow for cells' internal environment to differ from its outside environment. So selective permeability of lipid bilayers, specificity of proteins, and passive and active transport make a big difference on the internal environment of a cell. An evolution of membrane proteins enables early cells' ability to create this special internal environment, and thereafter, natural selection affected the efficiency in which these membranes worked. So membranes are very important. I love this summary diagram of actually diffusion, facilitated diffusion, and active diffusion. It shows the differences of each one. Remember, diffusion does not require any outside energy. And in diffusion, without a protein, you have the movement of small nonpolar molecules. We have facilitated diffusion to move large or charged molecules across a membrane. And then active transport requires energy in the form of ATP, which also begins with the letter A. And we're moving molecules against their concentration gradient. And this is the end of this chapter.